All right, so like I told you guys, I was gonna go ahead and record a review of sections 1 and 2 for you, kind of going over that physiology like I did with the rest of the sections. So let's just go ahead and kick this right off. It's not letting me maximize for some reason. There we go. Okay. And of course, it wants to repair. All right. We'll see how this goes. Oh, man. We might need to. We can go ahead and stop share. Okay, looks like we should be recording again. All right, got this pulled up. All right, or maybe not. Maybe it's not gonna work for us. Okay. Let's see, a couple things that I just want to get out of the way. I'm gonna make an announcement as soon as I'm done with this, but for the final, if I remember correctly, it's 75 questions on the lecture exam, multiple choice final. I think I've got, what is it? 2 of the essay questions like I've had. And then I think I've got, I want to say 8 of the multiple where you can pick any number of A, B, C, or D. And then I've got the rest, so that makes 65 multiple choice, I want to say. That sounds about right. So something close to that, but the outline is pretty much the same. You're going to have multiple choice, you're going to have multiple answer, and you're going to have a couple essay questions. So as far as the lab exam portion goes, because I stick to the same format, really all I'm going to be doing for you is I'm going to be taking the lab practical images, and Jess, looks like we got someone else. Hello. So I'm going to be just taking all those images, putting them into one quiz, and then again, you're just going to write down what you know. Look at the image, identify the structures you know, write them down. Obviously, I'm not going to have you know like the combination of all 3. You know, like I think each lab practical, you had to know about 150 points worth of information to get a 100%. You know, I'm not going to make you know 450 points of information to get 100%, because there's going to be about 1,000 points available for you. You're only going to have to know probably, I think I'm going to draw the line at about 200. So is it going to be more than an individual lab practical? Yes, but it's not going to be the combined cumulative of all of those. So honestly, and if you just know the names of your organs, bones, main structures, that probably will make up 50% of what you need to get. So you can get a 50% if you just study those major things, you know, major vessels, you know, of the heart, major, you know, nerves, muscles, and if that's all you studied, you would do really good on the final. So again, you know, concentrate on what you think is interesting, you know, and what I've marked as most important, because that's going to hopefully help you moving forward understand the best. Hopefully this, goodness, I don't know what's been wrong with my PowerPoint lately. It has not been wanting to work for me. So let me pull this back up so I can share it. Close. Hopefully All right. Okay. Okay. Close that. All right. So let's go ahead and just start right off. Should be sharing the screen. All right. Vocabulary, knowing anatomical position, pretty straightforward right there. Just being able to know your reference, directional words that you see in anatomy. Again, pretty straightforward. I'm not going to worry about those. These just, if I tell you to put your hand on your leg, right now, just touch your leg. You're probably going to touch your thigh. Really, in medical terminology, the leg is actually between the knee and the ankle. All right. So if I told you, however, to touch your distal anterolateral leg, then you know exactly where. You're going to go to the part farther from the knee, and then you're going to want to go on the front side and slightly to the side, to the outside edge. So you know exactly where that is. You've got a very good frame of reference. So that's what these words are for. Okay. All right. Body planes, pretty simple right there. These, again, most used when you're looking at imaging, MRI, CT, and MRI, CT, and X-ray is where you're going to be kind of just knowing how you're looking at the body regions and cavities, just dividing things up categorically. So just, again, these three are probably the most important ones to know, and ventral body cavity. But other than that, honestly, I'm never going to ask questions over this. I don't even know why I still have that in there, so don't worry about that slide. Okay, so mitosis. This is cell division in normal average cells in the body. All right, human cells have 46 chromosomes. All right, and they have a copy. So they have a copy of 23 pairs. All right, of each pair of chromosomes holds information for the same kinds of things in the cell or in the body. All right, and you get one from the mother, one from the father. All right, and when a cell has pairs of chromosomes, it's called diploid cells. All right, so the processes goes through. You've got prophase, metaphase, anaphase, telophase are the four phases. I explained these pretty clearly in here. So this again, this chromatin, chromatid, this is essentially that DNA when it's unraveled. All right, and so it's going to supercoil up into those little X shapes that you're very familiar with. And yeah, so then you're going to start to see these little mitotic spindles that are eventually going to attach to those chromosomes. All right, they're going to pull those chromosomes and line them up in the middle, right there at the metaphase plate. All right, and they're going to line up individually. All right, as opposed to in their pairs. All right. And so then these during anaphase, they're going to split. Now I remember anaphase from anaphase away. All right, is a good way to remember that. Anaphase, this is where your chromosomes, they start pulling away to the outsides of the cells. All these daughter chromosomes right here. All right. And then during telophase, you're going to see those chromatids, all right, they're going to unravel, all right, into their less dense chromatin forms, and then you're going to have that nuclear envelope form around into the nucleus, all right? And that chromatin, that unwound chromatin, is what there's going to be, you're going to have your RNA going to be able to go into the nucleus and copy it from there, all right? The chromatids, all right, that form, those are just a compact version that allows it to be split apart. All right, and then cytokinesis, those cells are going to start to pinch off right there. Okay, pretty clear. This interphase is honestly, most of the cell life is in interphase. All right, and so you're just during G1, you're going to have that cell growth. Then you're going to start to replicate the DNA, because again, when you finish off here, right, you have all 46, chromosomes, but there's only half, right? And if you split those again, well, you can't. So you have to replicate that again so you can go through that cellular process. So during S phase is when that DNA is going to get replicated, so that you can form the whole chromosome or the chromatid. And then G2 phase is just essentially final preparations, getting ready for that mitosis. All right, so as you can see here, this is a good representation of how most of the cell cycle is spent in interphase. All right, okay. Let's see, so sexual cell division. All right, so this is when your cells are gonna be getting ready to for sexual reproduction. All right, and so it'll result, instead of diploid cells that have, 2 copies of each chromosome, you're only going to have 1 copy of each. All right, and that's because the child is going to have a copy from the mother and the father, and so when that forms, then you'll have a diploid cell in that zygote. All right, the zygote is the name of the cell that's formed after fertilization. All right, haploid cells 23. So most of the process is the same, except I just highlighted here what was important. So you get homologous chromosomes form tetrads. All right. Okay, so let's go. Do I not have a picture? Okay, I guess I don't. So what you're going to get is you're going to get pairs of DNA. Man, there should be a picture somewhere. Let me see if I can find one real quick for mitosis, because I would like to mitosis. Jizz. All right, let's go ahead. We'll stop share that. And pin me. And then we're going to share right here. Okay. So here, as you can see, it is a little bit small on this. All right, but hopefully you can see there, you've got the red and the blue here. That's a pair. So that's chromosome 1, and you have the 2 pairs. And so that's the individual copies. And it's not all the blue and the red going on the one side. And the blue and the red is going to represent kind of essentially the chromosomes you got from your mom and your dad. Well, what the cell does, it kind of lines those up a little bit randomly. So one sperm cell, all right, or one egg might have 10 chromosomes that came from your mom, right? And then 13 that came from your dad. And so that mixes up that DNA a little bit from generation to generation. And then on top of that, you have something called synapsis, where these tips here, all right, they're going to exchange little bits of information. All right, so you'll get the very end of that chromosome. You've got 4 points on each chromosome, right? So it's gonna switch, and so that's gonna put a little bit of mom's DNA onto the dad's DNA. So you can see this really randomizes that DNA to give a lot of diversity. All right, so that's pretty much the purpose there. So I'm gonna go ahead. I'm gonna share. I think I can just do it from here. Share. All right. So we're going into different kind of tissues, the integumentary system. Okay. When you're looking at tissues, you're basically going to have your apical surface, all right, or the top, and your basal surface, which is usually going to be that underneath. layer. All right. Cilia are hair-like projections that are used like for propulsion, and your microvilli, they're smaller, all right, and those are going to be the folds in the membrane. So if I have my fingers like this, if you look in the smaller picture, all right, to the side, all right, you can see this is kind of that outline right there of the cell. But if I slip my fingers, I increase the amount of the length I have to draw that line, increase the surface area. So that's what microvilli are. And then cilia are little hair-like projections that cells can use to move around. All right. So these are just some general types. You're going to be looking at simple cells. All right. Simple means that there's just one layer. Stratified means there's multiple layers. Squamous means the cell is kind of flat. Cuboidal means it's kind of like a cube. Columnar means it's kind of rectangular and taller in shape. So if you just put these, together, you can know, oh, simple squamous, it's a single layer of flat cells. Stratified cuboidal is going to be multiple layers of cuboidal cells. And there's going to be an apical surface, which is facing the outside of something, and then that basal surface. And that's really going to depend on what you're looking at, what becomes the basal and the apical surface. So right here we have a vision of some simple squamous. These are the alveoli that you learned about, okay? And so you can see here, this is that alveoli area right there. And so this right in here, this is going to be that apical surface. And then in the middle here, you can see you've got like kind of two layers of cells going through, right? A little bit maybe, all right? Going through. And so they're going to have that basal surface in between, okay? Oh, let's undo that. Okay, these are going to be, and again, it's pretty clear where those are found based on that purpose substance exchange. Because it's only one layer of a very thin cell, it makes it really easy, it allows the process of exchange from one area to another, not just from the outside of the cell to the inside of the cell, but then again back outside the cell. It makes that process, it's able to happen a lot more quickly because it doesn't have to go through as much space. You get a cuboidal cell, and it's gonna take some time for it to diffuse through the cell wall, travel through the cytoplasm, and then diffuse out the other side. So these simple squamous, right, gives that that easy layer to diffuse through. All right, simple cuboidal. All right, now these cells are larger in part because they're going to be producing and excreting substances. So these cells are creating things, in your, they could be producing hormones, they could be producing enzymes, in the pancreas, in the pituitary gland, and then they're going to be excreting that. So right here, you can see this is kind of that apical surface, all right? And so we're looking at more of like a transversal cut, all right? So you've got this vessel that might be running, and we've cut it here like this. And so we're looking on top like that, down this tube, okay? And so in here, this is that lumen or the opening of that tube. So that's gonna be the apical surface, all right? And this will be the basal, all right? And I actually don't know where this is specifically. Could be anywhere that's gonna be secreting substances. So again, small ducts of glands. So this is some kind of gland in there, all right? And these will be used for, secretion, can be used for absorption as well, all right? And then again, columnar, and you'll see these a lot in intestines. This is where you've got a lot of absorption going on. You can see you've got this kind of top layer here. You see that little line, and then it's got almost like the ends are kind of fuzzy. That's all the, and of course it crashed again. That's where you have all the microvilli, all right? Or you've got villi and microvilli. The microvilli are on individual cells, and villi are folds of columnar epithelium. Open this back up. Pause recording, I guess. Resume recording. All right. Okay. All right, so let's see. Go through pseudostratified columnar looks pseudostratified because what you have is you really when you have stratified. Let's see if I can get. You can see the cells are literally the bottom of one cell is attached to the top of another. Whereas in pseudostratified, you actually have everything is attached here to this base at the basal surface. They all have the same basal surface, but then here, this one is just kind of being crowded out by these other ones in here. All right, and these are going to be found in the respiratory tract. These are not microvilli, these are cilia, really long cilia. All right, and they're essentially going to help. They're going to create a brush border, which is going to help to get phlegm out. So when you're coughing up phlegm and you have that really productive cough, that means your pseudostratified columnar cilia in the respiratory tract are working overtime, trying to clear out the lungs. All right. Keratinized versus non-keratinized. Keratinized means there's keratin, all right, which is kind of a hardening substance. It allows your pseudostratified columnar to harden. And the reason that you've got this stratified squamous, all these layers, right, means that if there's any kind of abrasion in the intestines or on the skin, all right, you can slough off a couple layers and then you just got tons more that can grow back. And then the keratinized, which is going to be found on the outside of your skin, creates an extra, toughness to those cells so that it can be a little bit more protective, and it's going to, keep things from getting in through those layers. Whereas, this is less protected. It's going to slough off a little bit easier. Things are going to be able to travel through these cells a little bit easier because they don't have that keratin to get through. That's kind of essentially hardening the cells in a way. Stratified cuboidal, you won't really find these much other places than mammillary ducts, all right, sweat glands, all right, where you got to produce a lot of stuff. So really think of the cuboidal as for secretion. You want to secrete a lot of stuff, produce milk or sweat. I mean, you're producing a lot of stuff all at once in one area. So that's why you've got lots of cuboidal cells in that area going on. Stratified columnar, just layers of that columnar, and you're going to find that sun duct's pretty rare to find throughout the body. Okay. All right. And if I were a student and looking, I'd be like, that's not really specific. I don't know that he'd ask any specific questions about that. Transitional. So essentially these cells they're really stretchy. So found, I don't think they're exclusively in the bladder, but pretty exclusively. I can't think of anywhere off the top of my head where else they might be found. But pretty much the only place you're going to see them really is in the bladder, and that's because it needs to expand and hold that urine. And then you have the stretch reflex. So once the bladder gets stretched to a certain area, it's going to send a signal to your spinal cord, and that's going to say, I need to pee, and it's going to contract that detrusor muscle that we talked about in the last section, and squeeze that urine out. All right. And so, these, you can see you've got these bulgier ones on top, these flatter on bottom. Okay. I mean, this is almost, some of these are like pseudo-stratified, where you've got lots of cells connected to maybe the bottom layer and they look like it's another layer. And then you do have cells connected on top of those. So, yeah. All right, glands. Now you're dumb. All right. How do I want to explain this? All right. So, endocrine glands. These normally they're excreting. Usually, it's like into a blood vessel, like straight into a blood vessel. All right. And then exocrine. Usually, these, think of. They're going into some kind of, man, that was really quick. I have no clue what is going on with my PowerPoint. I need to reinstall it. Let me pause that. Zoom recording. Share. Share. Go back down to where we were. Well, roughly about here. No. Glands. Okay. So exocrine is usually going to be into some kind of large cavity or onto some kind of surface. So can't think of any good examples off the top of my head, but usually it's going to be going into a large lumen that will then be emptying out into the blood at some point. All right. And then just knowing merocrine versus holocrine, you don't have to know. This is just showing that these are excreting them through some kind of diffusion or, going through the cell membrane. And these ones, these cells are rupturing and releasing their contents, kind of like a bomb going off. All right. Connective tissue. All right. So, connective tissue really has, it's hard to describe connective tissue as a whole because it has such a diverse amount of roles, but essentially it's connecting one thing to another, whether it's kind of connecting two layers of cells, two layers of skin together. You'll find tissue in between there. And it can be for the purpose of lubricating. It can be for the purpose of support. Lots of different things going on. All right. Or you have entire, your ear is made entirely up of connective tissue. And then of course it's got keratinized, stratified squamous, right, on the outside, kind of covering that ear, right? That's what you're gonna find out there. But it creates a framework, right, for everything. So it creates it's supporting, right? We need these curves are important for conducting hearing or noise, conducting noise into the eardrum. All right. And so again, forming a support structure there. So lots of different kinds of fibers and things that can be involved in connective tissue that you're going to find in different proportions depending on what you want. If you want something that's more stretchy, then you'll have a higher, constituents of elastic fibers. All right. If you want, something really tough, right, you're going to have a lot of collagen. All right. Or reticular fibers. All right. And then the fibroblasts and the chondroblasts are going to be secreting this ground substance, which is essentially just it's like the broth of the soup. You can think of connection to as like a big soupy mess, and you've got all these fibers in there, right? Okay. And then it's just in this broth. All right. And so the fibro and chondroblasts are going to secrete that. They're also going to secrete the different fibers that are in there as well. Okay. So these are just things that you might find inside connective tissue. All right. So you'll see white blood cells traveling through the connective tissue to get from blood vessels through to the body and to another organ or something, because this connective tissue will surround entire organs. And so in order for the body to respond more quickly, sometimes it'll go straight from one blood vessel, travel through the connective tissue, and then enter into and work its way between other cells and an organ. All right, to find bacteria that are doing the same thing and gobble up those, right? All right. So 3 general types. You'll see loose connective tissue, dense, and then cartilaginous. Essentially, loose and dense is just, with the loose there, it's going to be really brothy, lots of ground substance. All right. And then your dense is going to be have less broth. It's going to be more meaty. It's going to have a lot more of the fibers in it. Okay. And so these will be a lot more for, probably support type things right there. All right. Areolar tissue. All right, so they're, essentially these slides lay out a couple of the what makes up the areolar tissue. All right, and then so this is going to have again lots of ground substance. I mean, it's loose, right? I said so it's brothy. All right, and this is going to play that big role in inflammation. All right, because it's got all this ground substance. There's not a whole lot that macrophages, and white blood cells have to work through to be able to travel through. It can just fly right through that fluid. So it's going to play a big role in inflammation, and this is actually where you'll get when you get that fluid that goes into the lymph or out from the lymph. That like this tissue is really where all that fluid is going to cause inflammation. When you get an area that bulges, your areolar tissue is actually what's receiving all that fluid, all that water content to make it bigger, right? So yes. So you're going to find that around organs and tissues. All right, adipose tissue, pretty straightforward. All right, reticular tissue. All right, so this basically just supportive tissue that you're going to find around the spleen. Has lots of reticular fibers inside of it. Okay. And so really this is a lot of what this is doing, just kind of like holding those cells in place, really is what it does. It's kind of supporting those cells, keeping them in place. And that's really what it does. So now we're getting into this dense tissue, right? Super dense, not a lot of ground substance in there, all right? So this is what you're gonna find in tendons and ligaments, all right? And again, dense regular, so it's all in a line there, everything going in a line. It creates a lot of tensile strength, all right, in one direction. And so that's to create to keep your bones from pulling apart. You've got your ligaments, or to keep your bones attached. You have your muscles, they pull in one direction, and so there's no need for it to be irregular, which we're gonna talk about right here. And so this is where you've got kind of multiple directions of stress. So you think about a joint. At the end of a joint, you've got that little end capsule there, right? That's protecting it. And so you're gonna get, when I hit my hand like this, I need to be able to have this kind of direction. Or I could get hit from the side like this. And so there's lots of directions. And so it's there to, resist that tension in many different directions. All right. So that there is an omnidirection in those layers in the collagen. All right. All right. Elastic tissue, lots of elastic fibers. So you probably, understand now that our arteries, right, we have vasodilation and constriction, and so you need to be able to expand and contract without losing without getting stretched out and remaining that way. So these elastic fibers allow, once something's become vasodilated, muscles can relax and then come back together, right? Actually, I guess if it's coming back together, it's going to be the muscles contracting to constrict that. But you get the idea. It helps to create that elasticity. All right. Hyaline cartilage. All right. Support and reinforce. So you're going to find this as costal cartilage, end of bones, and around the trachea. It's very tough. All right. Okay. Elastic cartilage, all right, has more elastic fibers in it, all right, to allow for a little bit more stretch, all right, a little more, playability. You might, this cartilage, elastic cartilage in my ears, a little bit more pliable, right? Okay. All right. And fibrocartilage. So fibrocartilage, again, so it's a little bit more organized, kind of like our dense regular tissue. all right, with kind of fibers tend to run in one direction. And that's because, for example, in your discs, all right, it's like, that I think I might have described in a video. I feel like I said this, but anyhow, at one point, you've got, it's like a jelly donut, right? And it's kind of like you got this jelly donut is being squished on the top and bottom by your vertebrae. And so it's got, if this is the top or bottom, and you've got right here is your disc, right? You're going to be pushing on it down, right? Coming this way, okay? And then you're going to have forces trying to push out in all directions. And so these fibers are going to keep it in there, lock it in there, all right? And so when you get a disc bulge, what happens is you start to see essentially these fibers start to rip, and you get a tear that starts going further and further out. And then that little nucleus pulposus, that jelly of the jelly donut, is going to kind of start to squeeze out and stick into the spinal canal and can compress, all right, that cord that's in there. All right, epidermis cells. All right, so talked about keratin earlier. All right, so it's protective protein that for the skin, melanin, that's going to give your skin pigment. Yeah, don't worry about these. We talked about tactile Merkel cell. There are lots of different kind of nerve cells we went over. There's lots of different kinds. Dendritic cells. So what does dendritic mean? Dendrite is kind of like an arm. It's a cell with with the nerves. They have the dendrites, right? And so those arms that went out. So these dendritic cells can kind of reach out in through the skin, the epidermis, and kind of find things there and make contact with bacteria and say, oh, there's bacteria over there. I'm going to start crawling over and grab it and eat it, right? With phagocytosis. All right, different layers and their purpose. All right, pretty clear there. Just picture that. Dermis layers. Okay, so this is the layer underneath the epidermis. All right, it has two different layers with different purposes. All right, so this papillary layer is going to create smaller ridges, all right, like our fingerprints. And then the reticular layer is going to create more of the friction lines, these deeper fissures in our hand. Okay. So epidermis going through here, right? And then here are those other layers. So the reticular layer, you can kind of see it's got those reticular supportive fibers in there, right? Kind of support that, all right? And then underneath this will be muscle. So these layers of skin, right? As you get through, you'll have a layer of connective tissue, right? This reticular layer right here, all right? And underneath will be muscle. And what happens, that connective tissue is there to actually create, a place where there's where the muscles can slide up with the skin. And when you get things called adhesions or scar tissue that forms in there from damage from one thing or another, whether it's a cut that goes in there and forms damage, well then you can't move. And so it's like if I'm pressing really hard, my hand is, the layers of muscle and skin are just going to pull up against each other, and that's going to start to make the muscles sore and tight because they're working against that resistance. All right, hair follicles and nails. You can see I didn't really expect you to know anything of that. Just sudoriferous glands. So our sweat glands, all right, really just know that there are two different types, okay? Your eccrine glands, which you're going to find on the palms, soles, and forehead, and then your apocrine glands, all right? If I remember, oh, where are you? Okay, don't worry about it. All right. We won't go into the difference between the eccrine and the apocrine. Just know where they're located roughly. All right, apocrine glands. However, ceruminous glands are going to be found in your ear. All right, and they're going to make your earwax, and then mammary glands are considered apocrine glands. All right. Okay. Sebaceous glands. All right. Going to create sebum. All right. Sebaceous sebum. All right, and these are oil glands. All right, that are going to result in zits, blackheads, and they're all over your body. All right. Functions of the skin. Right here. All right. I don't know if I have to explain too many of those. What they are, the reason, right? Protection, physical and chemical, from damage from someone hitting you or scratching you. Chemical, could be spilling acid on yourself. I mean, that's an extreme example. And then biological, it protects against. I don't know why I don't have that one, but it's protecting against bacteria, right? Helps insulate your body. All right, is where our all those nerve endings are that are going to be sensitive to touch and heat and everything there. It's important in metabolic processes, right? Forming vitamin D, it's where that sun is going to come in. All right, come through. All right, and then excretion, sweat, oil. All right, won't have any questions. I need to delete this slide as well. Okay, go through bone types. Let's see, what do I want to say about this? So these long bones are really good for levers. So that's why we're going to find long bones in our appendages, right? Because they're going to be there, so the muscles can attach and have a lever so that we can move. These short bones right here, like the talus, are more for kind of fine movement type things, right? And regular bones have just different purposes, very complex purposes. And then flat bones are usually actually going to be for protection, right? The sternum is there to protect, right, your chest. The skull bones are flat bones that are for, going to protect you there. All right. Knowing different parts of the bone. All right. And 1/3H4 going through there. All right. So you've got this spongy bone in here that's full of holes. It's very airy. All right. So there's going to be lots of blood vessels going through there, in here, because as we learned that we've got in our bone marrow, that's where we're actually creating, all right, our blood cells. All right, and the yellow bone marrow, that specifically is fat storage, as a matter of fact. All right, okay, but we've also got red bone marrow. All right, compact bone is this protective layer right here on the outside. That's going to be a lot thicker. All right, or more dense, I should say. Diaphysis, epiphysis, just shaft and the ends of the bone. Periosteum is an outer covering. All right, it's full of nerve endings. All right, which is why you've got you'll experience pain when you break a bone. All right. Okay. And the endosteum is the inner lining. So I don't think so this is just the lining inside of the bone. That's, again, just another protective layer kind of to insulate this compact bone, or just the bone in general. All right. Let's see here. No bone marrow, fat storage. So cover that. These are just, again, are just words you're going to see as kind of themes going through. You're going to see tuberosities, tubercles. So, the X tubercle, the Y tuberosity, right? Just knowing what those are, knowing these words are going to help you to understand maybe where they're located a little bit. Tuberosity is a big kind of prominence. A tubercle is a smaller, tiny little prominence that you'll find on a bone. right? Epicondyle is kind of a little thing, a little ridge at the end of the bone, okay? And then a process is something that sticks out and is more prominent. It's, you know, coracoid process, right? So, and then these are different depressions over your fissures, foramen, notch, sinus, right? Fossa. You know, just gonna, you can look at those. Your foramen is gonna be a hole, right? Notch is pretty self-explanatory. Okay. Sinus is a hole inside of the bone, right? Like your sinuses. And a fossa is a big, large depression. Okay. All right. Let's see. All right. So the articular surfaces, this is where the bones are made to kind of come into contact with each other so they can move. All right. So you're going to find a layer of cartilage at the end of the bone that's going to protect that bone surface. All right. And keep it from having any kind of wear and tear. All right, and this articular capsule is going to cover that joint space to keep in this synovial fluid. All right, so you got the fibrous layer on the outside and then the synovial membrane on the inside. All right, so then it's filled with that fluid that is going to help to prevent friction in that area. All right, lubricating it like oil. All right. Ligaments hold that. Bursa or parts surrounding, these are going to prevent friction between parts surrounding a joint. So, you know, you've got all these muscles that are running across bony prominences. And if they were to contract over this, you know, you got this ridge here and it's contracting, you know, over this area over and over, that's going to rub it raw and eventually cause it to break. And so the bursa kind of gives it a little bit of space, a little smooth thing to go over, so it's not going to do that. And then the tendon sheath also is going to help with that. All right. So just here's an example of a bursa right there. Okay. Right here, this muscle is the supraspinatus tendon coming through. If you have rotator issues, that's a pretty common one to get injured. All right. If any of you sleep with your arm up like this and you've got issues with your rotator cuff, this is you know, one of the biggest predictors for someone having or it's, I don't want to say one of the biggest, it is something that is seen a lot in people with supraspinatus issues, all right, with their rotator cuff. They sleep like this because that is going to be compressing, even though you have that bursa there, it's going to be compressing that tendon, cutting off blood supply, leading to degeneration and eventually tearing. All right, so joint classifications, you can classify a joint based on the way it moves or how it is connecting. All right, what is making, is it fibrous? All right, using more you know, fibers, ligaments. All right, and then, or is it using cartilage, fibro or hyaline cartilage? All right, so this would be like your dense connective tissue. All right, that's fibrous, right? Okay. And then synovial is going to be that main one that we're talking about with you know, at your elbow, where you have a synovial cavity and a synovial membrane. So that's what's going to be connecting it there. All right. So 3 different types for each way of classifying it. All right. So the same joint can be a diarthrosis, freely movable, and a synovial joint. All right. As a matter of fact, most synovial joints are going to be diarthrotic. Few might be amphiarthrotic, but it totally defeats the point for there to be a synovial cavity that's going to allow for a lot of movement in a synarthrotic joint. Okay. So usually the ones that are going to be synarthrotic are going to be things like sutures are going to be synarthrotic because the sutures in your skull, there's really not a lot of movement going on there. All right. Okay. Classifications, you can see that. So these are different kinds of synovial joints for you to know. All right. The ball and socket is going to allow for lots and lots of movement. All right, pivot joints like the elbow is just going to allow that to pivot, supinate, pronate right there. All right, we've also on the dens allowing the neck to pivot. Okay, right there. Now most of the rotation in the neck, you got a lot of it coming from that dens. All right, but then you also have a lot of it coming from these plane joints. All right, well actually it's not. It's closer to, yeah, plane joints that are at the those transverse processes. Okay, where you've got these areas, all right, that are sliding up and down on each other as I'm extending my neck or my back, right? And we'll go over those bones. So these are just some examples right here showing you those growth. Different function of bone. I don't think I need to go over that too much. Self-explanatory. Bone cells and their purpose right there. So so when an osteoblast matures, it's going to become an osteocyte. All right. Let's see here. Lacunae. All right. So this is going to house the osteocyte. So essentially, it's like an osteoblast. All right. Is actually it's kind of gonna essentially it's like painting yourself into a corner. All right. It gets into that bone and it excretes all this stuff all around it, and then it's in this cavity and just kind of like sits there. It's like, okay, what am I gonna do now? And so it just kind of goes dormant, becomes an osteocyte. All right. So you know, osteocyte, it's in one site, right? It's stuck there. All right. And blast, think, you know, something blowing up, something's being created, I guess, you know, and then classed for being the bone resorption. All right. So knowing what these look like, I don't think I had those on the lab practical. All right. So this canaliculi communicate between the lacunae because these osteocytes, they can't die off. They need, you know, they're sealed in, but they need some kind of nutrients, right? So these canaliculi are going to allow for that communication between the lacunae. All right. The osteon is considered the functional unit. So it's going to be essentially this osteocyte and everything around it, all right, is going to make up that functional unit. All right. Okay. All right. So we have, oh, sorry, actually, I was a little bit wrong there. It's going to be based around the central Haversian canal, so what this osteon and the lacunae are going to be kind of around it in those the lamellae, all right, forming that. Excuse me, I misspoke there, all right? So you can see in here, you've got these little osteocytes, right? In here, you've got this central canal, right, creating the osteon right there. The lamellae are these different layers going around, you can see there around it, right? Those different lamellae. Perforating and Volkmann canals, these are again to allow communication between these different layers, between these different osteon, all right? Going through the bone. So as you can see, you've got, you know, a little blood vessel, all right? A little nerve, or two blood vessels, an artery, a vein, and a nerve going through this bone. So that's why breaking bone hurts so much. All right. Bone formation. All right. Got a couple different types. Chondral. This is bone formation that's occurring in young children. So you got this Highland model that's going to start to be created. Going to start to ossify, right? Kind of around that bony collar right there, right? And then it's going to start to that, right, matrix in the middle, right? Empty space where you're going to start to fill that yellow bone marrow, and red bone marrow is going to start to fill in, okay? And then the remnants are going to be essentially up top here. That remnant is going to become the articular cartilage. And then right here, that's going to be your epiphyseal plate, where you're going to start to grow and expand and lengthen that bone over time. All right. Okay. So long bone formation, again, after you've you know, finished you're ready to start growing in length, all right, you've got this layer right here. And so this is going to be a zoom in of that. See, now this is actually, having this here is a little bit misleading, and I apologize. This is actually how flat bones are created. I think I have it right. Intermembranous, all right, formation. So I apologize for that. Having that on this slide, that's a little bit misleading there. But this, so that's showing that process. What I want you to see is to see this happening here. All right. Okay. All right. So bone remodeling. So our bones are essentially kind of the storage place for calcium. It's where our body puts the calcium so that it can be released into the blood when necessary, right? We have fat cells are essentially where the body stores stuff so that we can release energy when, you know, we need. And sometimes we need to, you know, release calcium. And so that's going to be stored in the blood. So the osteoclasts, all right, are going to be stimulated by parathyroid hormone, all right. The parathyroid, there are a number of things are going to help to detect low calcium levels, and so that's going to stimulate the parathyroid to secrete parathyroid hormone, and that the osteoclasts have specific receptors for that, right? When we talked about hormones, so osteoclasts have receptors, and that's going to make them more active. All right, and so they're gonna get this little signal and they're gonna start releasing calcium from the bone back into the blood. And then an osteoblast is gonna come along when it's not needed, all right, and it's just gonna start putting it back. And so you have this balance of, you know, osteoclasts are gonna be, you know, constantly releasing calcium from the bone back into the blood, and the osteoblasts are putting it back. And you know, it's just that balance. And so when the PTH is stimulating and the osteoclast activity is greater, then you're going to be, you know, losing more calcium from your bones. All right. And then just going through the bones here. Anterior and posterior fontanelle, those are the, when you know, that soft spot on the baby's head, all right? You're gonna see that up front here is the anterior, and then back here is the posterior, all right? So there's actually two of them on a baby, and if I remember, it's that posterior one closes before the anterior one. So on here, so we've got our articular processes, all right here, which I think I called them transverse processes before, which was wrong. So articular process right there, right? Okay, where you're gonna have that sliding, that's gonna allow for extension, rotation, okay? All right, so superior facet on the inferior facet, create that movement. Okay, this atlas is gonna hold up that skull and then sit on top of the dens or the axis, which is gonna allow for that rotation, okay, in the head. All right. All right. The articular pillars, that's just the word for, let me see if I can find here. So it's kind of tiny right here, but you see, I wish there were a better picture. A little bit, you can see it a little bit better here. So you see here as you go through, you can see where all these articular processes line up. Okay, it's kind of like a ridge, but then once you get up to here, you see it's almost like a big pillar. All right, that's so you know, it looks like just this block, right, going straight up. So the articular pillars are referring to the, you know, kind of that structure. Let's see if let's go up here. It doesn't show a good example here of from the side, but a cervical vertebra has kind of that individual part of the pillar that is just there, kind of like a little mini column, and you stack those on top of each other to get the articular pillar. But the articular pillar, it is actually talking about the individual part of each bone, and then it creates this large thing right there. All right, so the articular pillar is not referring to that whole structure of the pillar of this ridge created by all of them. The articular pillar is actually referring to, it's essentially right here, right? Looks a lot like a column, all right, on each individual cervical vertebra. But you know, that's not really that important. So I don't want to confuse you by going over, you know, these smaller ones. The mammillary process, you know, is just these little pieces right here, this little edge kind of back here that's hanging off of the articular process. All right. And, you know, back here by the transverse process, it's kind of through there. It's not even listed on here really. And then the pars, this is going to be kind of coming across right in here. Well, sorry, the break is going to be there. So yeah, this is considered the pars interarticularis. So this connection between this superior articular process and that inferior articular process right there. This part is breaks often in the low back. You can get a stress fracture there just from, you'll see this often in gymnasts from their hyperextension, right? It's just going to kind of wear away, and that bone will get kind of weak over time, and it'll break right through there. And so, you know, you'll have a break in the pars, is the way we say it. All right. Ribs and their parts. All right. I think that about does it for the bone. So I kind of forgot. I felt like, man, I felt like I've told you guys this before, and I think I did do a kind of a review after the test. Although I got to say, I don't remember if I recorded it. I think I did, but I also don't remember if I went into a whole lot of detail on it. So hopefully this, going into a little bit more detail, helped you guys. Otherwise, yeah, no one's here to ask any questions, so I just won't worry about that as I go through. Yeah, all right, that's it. So I hope, I think this will be the last thing or whatever for the class. You know, I've enjoyed you know, being able to teach the class. I guess I can't say like I've enjoyed being with you this semester because I haven't actually been with you, but interacting with you has definitely been an enjoyment. You know, I do consider, you know, I enjoy teaching. I consider it a hobby for me, and because I enjoy it. So hopefully I've done a decent job. Again, moving to the online format was a little different. You know, my main goal is to help you understand the content. And be able to set you up for success in your future careers, whatever they may be, whether or not they're related to anatomy and physiology, but to give you a foundation to build on, so that when you're learning more complex things, you can look back on this anatomy and physiology and just say, oh, okay, that makes sense. You know, it's a lot of this knowledge doesn't always get used. You know, I don't talk to patients about their blood vessels. I don't, you know, I talk about the sciatic nerve a lot. I talk about, you know, the vertebral nerves a lot, but, you know, some of the nerves of the body, the muscles, understanding actions, you know, that's very pertinent to my profession. But knowing, you know, the complex processes of hormones and their effect on the brain, you know, that's, if I feel like there's an issue with that, that's where I refer out to someone who's a specialist. So, but being aware of what's going on, so that when a medical professional talks to me and is talking about, you know, oh, the limbic system is, you know, being affected by their hormones, I'm like, oh, okay, I understand that because I know relatively how the limbic system works. I know how hormones work. And so I know what you're telling me is oh, so is the pituitary gland going to be involved in that? And they might say, actually, it's more coming from the centers in the midbrain. It's like, oh, okay, yeah, there are also some areas in the midbrain that produce hormones. And so just being able to relate to professionals and that's really what this class is more about, so that you can have a common ground to build off of, even if someone knows more than you on that topic. So that's why, hopefully my testing with the labs has given you the opportunity to realize, okay, what is important for me that I really want to concentrate on, and then just learn these other elements so that I can be aware of what they are. So when someone talks to me, I sound competent, right? You don't want to be the nurse or the medical doctor or the physician's assistant or a veterinarian that someone's talking to and mentions the hypertrophy in the left ventricle, and you're just sitting there like, where's the left ventricle? Right? You don't want to be that guy. Yeah. But good luck on the final. So any questions, shoot them my way. Again, when you open up that lab practical and you see that there's like 100 different items to look through, again, don't get overwhelmed. You're probably going to have to get just 200 points to get it. I think I'm going to give you 2 hours on that one instead of 90 minutes because, 150 in 90 minutes and then 200 in 2 hours. I mean, that makes sense to me. So again, just put down what you know, concentrate on what you know, but study things, know the bones of the body. If you just knew the bones of the body, if you know the organs, if you know the nerves, the blood vessels, parts of the brain, all right there, you're going to get so many points. And that's not even brushing on individual parts of the adrenal cortex and adrenal medulla. Just knowing what the adrenal glands are, that's going to get you a point. Knowing what the kidney is going to get you, more points than knowing what the cortex of the kidney is, or knowing the cortical radiate artery or vein, right? Isn't going to get you as many points as knowing what the kidney is, because frankly, it's a lot more important that you know what a kidney is than the cortical radiate vein or artery of the kidney, right? So, just remember those are going to be keys and as you study.